Good morning, everybody. Hi, if I can get everybody's attention. I'm filling in for Muzz this morning, who unfortunately couldn't make it this morning. So I wanted to just start with some opening remarks. We had an amazing day yesterday talking about how we're going to take all of the data that we're generating, integrate it into the first international maps, and move those forward to release. All of the different bionetworks are actively working. We learned about phase 1 integration efforts, where we had a number of pilot projects figuring out how to integrate data, and now we're moving to wave 2 with additional body systems coming online to work on their integration efforts. All of those are being supported by amazing resources that are being provided in the community, data coordination portals and data wranglers, and these teams are really coming together now. So it's very exciting to see that connecting with ontology developers and all the different parts of the system that's needed to work towards these global maps that are international in representation and can be shared publicly. Today, we're going to think more about future directions. We're going to spend a lot of time hearing in talks and also participating in breakout sessions around two major topics. One is a lot of interesting information about new dimensions. Spatial transcriptomics is a big part of what we'll talk about today, both experimental and computational, as well as very exciting sessions, for me at least, about machine learning. There's all sorts of new machine learning technologies that can take massive amounts of data and do, have emerging properties come out that are very surprising, and we don't know what that's going to lead to in single-cell genomics. We've seen that with language, human language, but the language of biology will be interesting to analyze as well. So we look forward to all of these discussions today, and we're going to move first to a session where we are going to talk about spatial methods. And so I'd like to start by welcoming our first speaker, Fei Chen, who is coming to us from the Broad Institute.
Great, thank you. Thank you very much for the introduction, and thank you for the organizers for inviting me, and I'm excited to make it, despite the many flight delays. And just as a disclosure, I'm a founder in Curio Biosciences, which makes some of the is distributing some of the technologies that I'll be talking about. And as the introduction said, we'll be talking about spatial methods, and especially a new spatial method that my group is developing, and let's just jump right into it. Okay, so it's been really exciting to hear about some of these atlases being put together, and there's really kind of, at least in the beginning, two fundamental goals that many of the atlases are trying to accomplish, right? One is systematically catalog cell types and states within different types of tissues. And then another goal, as stated, is to map those cell types within tissue architecture. And so how are we doing that? Okay, so how to construct tissue atlases. If we go back to the 2017 white paper, this is figure 3, where the plan for constructing tissue atlases was put together, where there was an idea of having two arms, one for single-cell sequencing, where we would collect the cell types and states, and then another arm where you would sample the same tissues and collect spatial transcriptomic data to place those cell types and states back into space. I think for many of us, that's what we've been doing, right? In my group, in collaboration with Evan McCoskey's group, we try to apply this strategy actually for an entire atlas of the mouse brain. And in effect, what you do is you collect, basically using commercial sequencing technologies, really large numbers of single cells to form like the cell types and states scaffold. And then with spatial transcriptomics technologies, we have been generally collecting a technology which captures gene expression, but not at the single cell level, but at the near single cell level, and then placing those cell types and states back into space. Now, while this works, it provides some challenges. One is that there's these two pretty different workflows and two pretty different types of data. And the thought here is that we really like a technology or an approach that might allow us to have our cake and eat it too. Like, can we collect the atlas data as the cell types and states, by a single cell, and then still know where those cells came from. And so, we collected this data about 2 years ago, and today, in that time, we've been working on an approach called slide tags, which allows us to kind of complete the picture here for being able to collect both types of this data at the same time. And the idea here is pretty simple. Slide tags effectively allows you to do single nucleus RNA sequencing, and at the end of the day, it lets you place those cells back into space at 10 micron resolution. And how that works is that you take your tissue and you put it onto a spatially barcoded array. And in this array are a bunch of beads that have unique oligonucleotides at each position. You section your tissue onto that array. And then now, if you are familiar with Visium, what happens, or other spatial transcriptomics, what happens is you capture RNA here onto the array and barcode it. But instead, what we're doing, instead of capturing the RNA, we actually photocleave oligos off of this array and diffuse them into the nuclei, which allows the individual nuclei to be barcoded. Subsequently, we can isolate those nuclei and then perform any sort of single-cell, single-nucleus RNA sequencing. And so now you can think of these beads as like satellites, right? And the satellites are like sending out signals for how close they are to each bead, and the cells are picking that up. And so let's just illustrate what that looks like in a tissue. So here we performed it on mouse hippocampus just because it has a very stereotyped architecture. And here you can collect gene expression data, right? That's cells by genes. It's just standard single cell data. You can go ahead and cluster that. Here it's just like showing major cell type classes. Each dot here is a cell. But if you look here instead and look at the spatial barcodes, so like the satellite sequences captured by each cell, you see this green cell here has captured a bunch of satellite sequences from x equals 2000 and y equals like 1200, for example, right? And so what's effectively happening then is that we can go and fit the location of that cell actually from the distribution of barcodes that's captured, and then you can get a set of XY coordinates. And so here you effectively can place those cells directly into space. This is not like this is a computational reconstruction, but it's quite faithful to the architecture of the tissue. Again, now this is single nucleus data, but you can reconstruct basically spatial gene expression patterns. It recapitulates the tissue architecture, and effectively you have the same quality of data as single nucleus RNA-seq. And there's this interesting property of the data, which is that if you imagine these gray things are the beads, and the numbers represent how many signals each cell captured from a given bead, and let's say your cell landed there, it's actually capturing a distribution of barcodes, and if you fit the distribution, your standard error on that measurement is just something like the standard deviation over like the number of counts, the square root of number of counts, right? And that's actually very similar to super resolution microscopy where you have photons. So it's a nice analogy to imaging. But in effect, you can see that basically the standard error in localization of cells is actually much less than 10 microns based on the data. Okay, and then the other cool thing is that the data quality is virtually indistinguishable from single nucleus RNA-seq by both cell type proportions. Here, this is paired experiment where we ran one section on slide tags, which is like the spatially placed one, and one section on single nucleus RNA-seq. You can see that the cell type proportions are the same. The mean number of molecules or genes captured per cell is the same. And the normalized average counts within any given cell is virtually the same. And that kind of makes sense because you're measuring the same tissue with the same measurement modality. There's no intrinsic platform effects. And the barcoding step takes about 7 minutes and doesn't really significantly affect the nuclei quality. Okay, and I think there's this other aspect to it, which is that the single nucleus data has some pretty distinct features, such as really high molecular sensitivity. This is, on the same tissue. This is all generated on the mouse brain. You can see that, single nucleus data has on average maybe 10,000 molecules per cell when you sequence it to saturation, or maybe not even to saturation, but when you sequence it at high depth, and maybe an order of magnitude higher than spatial capture approaches, actually. And another kind of interesting effect that you see is that there's due to both the unbiased nature and the higher capture efficiency, you kind of generally see a lot of higher information in the data. And this is kind of an interesting way to visualize it. If you just look at PCs, the standard deviation of PCs across cells, you see that basically the slide tags data has a lot more variance in the first couple PCs, but also it has a lot more variance all the way out. And that's, I think, partially because if you look at pixel-based capture methods, you get intrinsic mixing between nearby cells, and that loses some of the variance. And in basically imaging-based methods, there's segmentation, and or in Xenium case, there's much fewer genes, right? So you're capturing less information. But I think this plot is actually kind of an interesting way to grok at that sort of intuition that we get about the data. Okay, so the takeaway is that with slide tags you can do basically single nucleus RNA sequencing, and then you can. The beauty is that we built a lot of experimental and computational workflows actually for doing single cell. Many of you guys in this room have been doing that, and that involves a zeroth order looking at RNA. But there's a lot of already existing workflows for looking at TCR sequences, copy numbers, chromatin accessibility, and there are other multimodal measurements, and in the future, potentially DNA and other epigenomic measurements. And so in our paper, which is on bioRxiv, we've actually looked at quite a lot of different test cases, all in human, including healthy human tissues like the tonsil and the kidney and the human cortex. But I'll just walk you through one case of data that we collected with slide tags in the case of actually a non-healthy human sample, a metastatic melanoma sample, where we actually did multimodal measurements of both RNA, ATAC-seq, as well as CNVs and TCRs. And so here we collected profiling on this piece of tissue, metastatic melanoma, and this tissue has this interesting histological architecture. If you look up at the H&E, there's these two tumor compartments that's separated by kind of this fibrous stroma. And you can see that first we performed slide tags on that 3 millimeter area there, and you can see that we have very high quality single nucleus RNA-seq resolution of these high level subclasses. You can place those cells back into space, a little hard to see, but if you just look at the tumor cells, these 2 or 3 tumor populations are transcriptionally distinct, but importantly they're also like spatially distinct. They separate out into these two lobes. But also you can see that tumor 1B population has some sort of spatial clustering within the tumor 1 lobe. And there also, if you use inverse CNV, this is like the x-axis of this plot is the correlated gene expression across genomic bins. So higher correlated gene expression tends to mean chromosome gains, and lower means losses. So there are some shared CNVs between the tumor 1 and tumor 2 population. You can see that's boxed there, and 7, and I guess that's 10. And then they also have some private CNVs, right? For example, chromosome 6 loss here in the tumor 1 population. And I think if you then do some neighborhood analyses, you can see that they also have distinct cellular neighborhoods. For example, CD8 T cells are strongly enriched in the near tumor 1 cells, tumor 1 population cells. And so you might ask why that is. What are the T cell states and receptors in that neighborhood? So you find that the CD8 T cells in the tumor 1 neighborhood are more cytotoxic. Tumor 2 neighborhood are more cytotoxic. They have higher enrichment of granzyme B. And then we can actually go and do TCR recovery. Here we actually recover TCRs that are paired from about 70% of the T cells, CD8 T cells profiled in this population. And you can see that there's this relatively large clone in blue that actually is paired, and it has a very distinct spatial localization between these 2 tumor regions. So this is a spatially mapped T cell clone that has a strong preference for one localization with one like subtype or like transcriptionally and genomically distinct tumor population. This is like pretty interesting. There's like kind of like, there's, just for the interest of time, there's like kind of like 2 reasons for this. One is that basically we find that tumor 1 has a strong depletion in antigen presentation and a loss of chromosome 6, which is where the HLA locus is. That's one hypothesis. And then we also performed some epigenomic sequencing, spatial multimodal sequencing with the 10x multiome that we haven't shown here. And we find that actually tumor 1 has this loss of melanocytic identity. So there could be a loss of specific antigens that this T cell clone is profiling. Okay, just to summarize, slide tags is like a single nucleus spatial platform that has 10 micron resolution in placing the cells, but the cell data is actually just single nucleus resolution. And it plugs into basically existing single cell workflows. All the data that I showed here was on 10x, but there's no reason why it can't be on Fluent or PipSeq or other technologies. And in collaboration with Curio, we actually profiled a very large region of the human cortex, a 1 centimeter by 1 centimeter region. And you can see that data here. And I won't have time to go into this, but this is some of the most high quality and largest regions of human postmortem single nucleus spatial data that's ever been generated. And looking forward, I think there's, if you're collecting single nucleus data, there's actually probably no downside to tagging it, right? Because it's the same data quality, and now you get spatial locations for about, sorry, I didn't mention this, for about 60% of the cells that you sequence, you'll have a spatial location. This is like my slide tags. It's an air tag on a slide. Okay. And I think going forward, I think there's really a lot of ideas about how you can leverage this to make really like multimodal measurements of tissue composition and dynamics. Thank you for your time, and happy to take any questions.
Great.
There's
Any
a lot
time
of
for questions? Yeah, so we'll do there and then there and then there.
Hi, my name is Bo. I'm from Genentech. Fei, so really awesome presentation. I had just one question about these slide tags. So is this for fresh samples, or you also do FFPE?
Fresh frozen samples.
And is there any plans for FFPE in the future?
Yeah, I mean, I think so. I think, FFPE is interesting. FFPE is almost always going to be targeted, right? And, one of the beauties of this approach is that it's untargeted. And we can get actual raw genomic sequence. So there's this trade-off. Yeah. But I think, the way to do FFPE would be through targeted measurements.
Yeah, thank you.
And one more question over there.
Oh,
Okay,
same question. There's a question.
Okay, is there another question? Yeah. Fabian over here.
It's really nice to have potential then coordinates as well, right? And I think your trick to go sub 10 microns was that you sort of have these satellite diffusion type of information. Have you thought about using also microscopy or maybe if you sort of do have this H&E sort of giving you additional information about potentially better localizing?
Yeah, that's a great question. I think that's a good insight, and definitely, it's very doable, I think. Yeah, I think, you first take a picture of where all the cells are, and then when you map it, you can like even probabilistically map. And you lose some cells in 10x and also in the nuclei isolation, right? So you could imagine also using the microscopy images to kind of do inference on the cells that you lost. Yeah. Okay.
Another question over there.
Yeah, wonderful talk. So I wonder for the TCR clones, did you actually do the TCR sequencing, or is inferred from the RNA? If it's inferred from RNA, is it the full transcripts or like the 5' or 3'? Thank you.
Yeah, that's a great question. We didn't sequence the genomic DNA, but we sequenced the 5' of the RNA that encodes the TCRs.
Inferred TCR?
Inferred TCRs. I mean, we sequenced the CDR3 region on the mRNA, but it's not the same as it's not sequencing it off the genomes.
I had one quick question. Have you considered multinucleated cells at all in any way?
Yeah, that's a good question. We haven't, and obviously when you do dissociation, there's a question of whether those end up as doublets or maybe you can tune your dissociation. Yeah. You can do it. If they separate, then you place them into the same location. That's nice. Yeah.
Yeah, and just a reminder to introduce yourself for also the virtual audience when you're speaking.
Thanks, Gary. It's Bruce Arno from Cincinnati Children's. Amazing. And that maintenance of the high variation out into large, high PCs, I think I agree with you, that was beautiful. And so could you harvest from that? What can you harvest from that that is, have you seen spatially specific markers in context that are coming out of that kind of thing?
Yeah, definitely.
Sub-perivascular specializations.
Yeah, no, I'd be happy to talk about that. I mean, I think that's a longer discussion, but I think definitely you can find spatially varying components. But I think the other thing that PC plot is telling us is what our intuition always sometimes tells us is that when you just do de novo clustering of single cell data, you can get much finer resolution on subtypes of cells than oftentimes you can get with spatial methods. And so it's capturing that information as well.
Great, thank you, everyone. I think we should, any online topics, questions? Okay, so I think we should move on. Thank you very much, Fei, once again. So we're now moving to Nadav Yayon, who comes to us from the EBI, and he will be telling us about a spatial thymus human map.
Hi, everyone. Okay. So my name is Nadav, and it's my first time at the HCA meeting, so I'm really happy to be here. And thank you for the organizers for giving me the opportunity to present this work that we've been working on over the past 2 years, which is constructing a spatial thymus human cell atlas, which is projected to a unidimensional tissue axis, and I'll explain exactly what this is. So the thymus is the best organ in the body, and I think we can all agree on that, because it's in charge of production of T cells. It's situated above this organ that we call the thymus perfusion pump, or the heart, like some people would say, but, and it actually slowly degrades after puberty like that. The most active phases of the thymus happens during fetal stages and then early pediatric stages of life. In terms of the architecture of the thymus, it's actually subdivided into these lobular structures, which are surrounded by this capsular membrane. And then these lobular organizations are further subdivided into the cortex, which is the higher density regions, and the medulla, which is the lower density regions. So T cells actually migrate through space through this process that we call migration through, maturation through migration. And kind of like the classical model of the thymic lobule indicates that the cells enter through this junctional region between the cortex and the medulla. Then the cells go through positive selection through the cortex, where they rearrange their TCR, and once they form a functional TCR, they then migrate from the cortex back to the medulla to be kind of like screened, so to make sure that they're not reactive to self proteins. So our motivation here was to see whether we can actually map these cellular processes and these interactions that these cells undergo in kind of like a continuous space, and also generate a common coding framework for this amorphic structure of the thymus lobule. And I'm not going to talk about the specific atlas perspectives here. Well, just to give you like a general overview of the data sets that we have acquired in this spatial atlas, which is mostly like a product of an amazing collaboration of a kind of like a centralized CZI thymus network, which involved like 3 major labs, which is our lab with Veronica, Lina in the Taghon lab, and Andrea in the Germain lab at the NIH. And we accumulated data, spatial data from both fetal and pediatric, so the most active stages of the thymus, both in terms of kind of single cell resolution protein multiplex through IBEX or rare site, and then also Visium spatial symptomics paired from these 2 time sets. We also accumulated quite a bit of single cell information and added new modalities to previously generated single cell atlases that were published by our lab, like the previous thymus cell atlas, which is really great. So how do you actually construct kind of like this model of this very, like I said, like this amorphic structure. So we said, okay, so we want to represent the highest degree of spatial variability in this structure, which is the thymic lobule. So for that purpose, we wanted kind of like to draw a line or a unidimensional axis that goes from the capsule through the cortex and into the medulla. But there is actually very specific and also non-specific challenges specifically for the thymus. So first of all, there is a very high degree of biological variance between the fetal and the infant thymic stages, as you can see in this image here. And in addition, we all know that we're working with human samples, and these are often very, very difficult and very large, and they tend to, especially the thymus, which is one of the most cellularly rich organs in the body, is sometimes difficult to section. So you often get like these freezing artifacts, or you're only sampling like a bit of the tissue, and you also can have like tissue tears in these things. So you want your model to be robust. The third component is the fact that we don't have 3D. So people have talked about this throughout the conference. And our assumption here was to say we're actually not aiming to do 3D, but we will want to minimize the error, the 2D error in sampling 3D space. So the way we did, and I'm going to go through this quite quickly, but again, if anyone has questions and these things, I really would like you to come and look for me, and we can have a further discussion about especially how we build this model. But generally, this is a nonlinear boundary landmark model of the thymic lobule. So in essence, it's centered around the position of the cell along that boundary. So by actually measuring the position of that cell in respect to its nearest neighbors in all the different macromorphological structures, we can position the cell along this axis. And actually, we're here to construct what we call the corticomedullary axis. We're combining two axes, one that goes from the cortex to the medulla, and another one that goes from the tissue edge or the capsule into the cortex. And actually, for the sake of kind of like, we were kind of like inspired by the biology of the thymus and by the fact that we wanted to capture kind of diffusion-like or cytokine-like effects. And because of that, the function that actually kind of represents this distance is a nonlinear sigmoidal-like function that gives you more resolution and thus more robustness around the boundaries between these regions, because you also have much higher spatial certainty of your position around these boundary regions. So this is how it looks like, and I'm sorry for the color scheme. I couldn't find anything better for that, but I'm going to show you better representations later. So on the left, you can see the actual annotations of the cortex, medulla, and the boundaries, and also like this gray, which indicates like an area that is not defined. And what you can see, I hope, that this model is actually quite robust to edge effects, to artifacts, and to tissue tears, because it accounts for multiple macromorphological factors to determine the position of the cell. In addition, so for better interpretation of the axis, we bin the axis into compartments. So very similar to what you would have, I would say that any common coordinate framework, you need to kind of be able to consistently map the same anatomical regions whatever tissue you're kind of mapping to this axis, similar to like the mouse brain atlas and these things. So we defined kind of like bands of region-like of the thymus, and then over the thymus lobule. And what we actually saw, again, is that for example, in lobules where we don't have a medulla, so it's very difficult to construct like the boundary between the cortex and the medulla, we can still account for, or minimize, like I like to call it, like minimize the 2D error and get closer to the true 3D space. And the analogies that I like to use in that respect is the analogy of the egg. So I all imagine that we're kind of like egg experts, and we can all kind of like, if we take a section through an egg, almost every human on the planet can tell us exactly, like, or with high certainty, where you section that egg and which angle, because we're all kind of egg experts. We've seen eggs like 1,000 times, much more than 1,000 times. But so in our representation, kind of like the thymus lobule is actually an egg, and the yolk is kind of like the medulla, the white is the cortex, and the shell is kind of like this capsular region. So because the model is actually scale invariant, it can handle very well differences in the absolute size of the structure. And here you can see a comparison between pediatric and fetal, which we can account for the same regions. But again, some of the regions, because they don't exist in the fetal, like for example, very deep medullary structures, will not be represented. So here I'm showing you actually all the data, so, which I think is actually important. And you can see like a PCA projection of each one of the individual Visium samples for the fetal and for the pediatric, color-coded by this axis. And as I hope you can see, but we also quantify this, the axis actually represents the highest degree of variability in the first and the second principal component in all our data sets, besides maybe one sample over there, which we need to kind of further investigate what went with it, but it could be like a technical thing. In terms of batch-corrected space, we see the same thing. And here I can also show you the comparison between, for example, IBEX. So the fact that we're annotating the image, like the basic, means that we can apply this model on any spatial modality. So we're not limited in that sense. And when you model, for example, 40-plex or 50-plex protein multiplex imaging, you actually get the same representation in the same reference space between all the different modalities. And then going back to our kind of like original intent, we used cell truncation, which is a great cell deconvolution method for Visium, and projecting back on kind of like this CMA space. We're actually quite surprised to see that the fetal thymus and the pediatric thymus are actually quite similar. And actually, in retrospect, when we went back to kind of like JP's work, we actually were not so super surprised, because we saw that the proportion of these alpha beta T canonical T cell trajectories, or proportions in PCW 11 up until age 10 are actually quite preserved. So the fact that we could actually correlate and say, okay, we see that the cell proportions are preserved, but now we can see that the actual basic tissue architecture is preserved, although the actual morphology is very different. So what I find the most exciting, let's say, about this project is now we can actually measure directly like the cytokine, the kind of like the cytokine highways, if you may, that kind of like span the thymus, and kind of like what the T cell actually sees while it's migrating through the tissue. And we perform kind of hierarchical clustering to identify differentially distributed gene groups, and found like specifically, like again, you can see that there's a high degree of similarity on one hand, but on the other hand, we can actually pinpoint what are the key differences between the fetal and the pediatric thymus. So to make, I'm not gonna talk about all the software packages that we've developed, but please reach out, and I would love to tell you much, much more. This was done in collaboration with Oren, a good friend of mine from the Anderman Lab in Harvard, where we developed a new pipelines and also software that you can annotate images directly into Jupyter notebooks, which is actually very useful for that purpose. And with that, I don't know if I have time to summarize, but this is just only one aspect from this very large project, where I showed you kind of like how we used the axis to map the global distributions of cells across the tissue and compared with fetal pediatric, but we also have obviously the stromal counterparts, and we also have some nice observations about CD4, CD8 migration timing through the cortex and the medulla, and also the organization of the Husserl's corpuscles, specifically in the medulla. So with that, I'd like to thank all the amazing people that really contributed to this study. And it's really been like an amazing group of people and the best people I've ever worked with, especially like Sarah, John, and Virginie, which were really amazing. Obviously the donors and their families, and all of you for listening.
That was great. We have time for one or maybe two questions. Any questions from the audience? I think there's one going back there.
Hi, Hari Nakshatri from Indiana University. I'm just trying to figure out how many epithelial cell types did you find, and have you found any correlation to thymomas? That's number one question. And do you see any distinct bacterial population in your studies, whether you have seen any bacterial population?
I didn't get the second part of the question.
The microbiome in the thymus.
Okay.
Is it distinct?
So So actually, like the goal of this project was to generate a healthy reference thymus. So we specifically did not look at any thymomas, any aging thymus, any kind of involution, and any of these. But we believe that this is actually like a baseline, that you can then go and see what happens around thymomas, what happens to these cytokines. Like, do you have like specific like influence that is going on there? And now you can actually like refer back to what you would expect to see in the fetal and the pediatric thymus.
One question from virtual.
Hi, we've got a question from Dana. Given your beautiful continuous approach, why did you then bin the data?
Because in the end, you want to, like the point about a common coordinate framework is that the community can now use something as a reference. So when someone, because in the past, if someone said subcapsular, nobody quantified where the subcapsular starts, where it ends. Like when people said junction, there was no indication if it's a junction. Actually, we believe that the term junction in the cortex might not be completely accurate. Like because there is the junction itself and there is the vasculature that is in the junction, and these are two different things. For example, macrophages are very prominent in the junction, but actually the entry point of the cells and the exit point of the cells is probably like we see more in the vessels in the junction. So it's very important to have like a common nomenclature and a common reference in that sense.
Yeah. I think we need to thank our speaker and move on. Thank you. So we're going to bring our next speaker in virtually, Jian Ma from Carnegie Mellon University. Are you ready to go? Okay, thank you.
Hello. Can you all hear me well?
Yeah, we can hear you.
Okay, thank you. Thank you for the organizers for having me speak in this meeting. This is actually my first HCA meeting, and I was really looking forward to attending in person and meeting many of you. But on Sunday, Air Canada canceled my flight, and they rebooked the flight right before this session today. So I'm sorry I have to present the talk virtually, but I hope what I will be talking about will be of interest to you and also the HCA community in general. So my lab has been really interested in developing computational methods, especially machine learning methods to study what we call spatial epigenomics, or this multi-scale spatial epigenomics, in different scales. From the spatial organization of the genome, the chromatins are spatially organized, to other subnuclear structures within the nucleus, interactions between chromatin and other RNAs and proteins or nuclear bodies, and even the entire cellular structure. This is a recent work from the Allen Institute for Cell Sciences to map the different organelles in the cell. And we also have tremendous amount of interest in modeling cellular spatial organization in complex tissues, which is something I would focus on today. But ultimately, I think our long-term dream is really to have a very comprehensive understanding of this multi-scale epigenome organization in single-cell resolution. I think Aviv already mentioned this yesterday during the panel discussion, but I just want to stress that again more explicitly, as the title of this Oscar-winning movie this year nicely summarizes our goal. When it comes to single-cell biology, I think we all hope that can achieve everything, everywhere, all at once. So in the past couple of years, my group has been very excited about bringing the 3D nuclear organization analysis to single cell level. However, single cell 3D chromatin analysis is tremendously challenging, and the data is much more sparse than other single cell assays. And so in this work, I will very briefly go through. This is from a former graduate student in Roche who is now at Broad. He developed a really exciting method to tackle this challenge. So the computational idea here is to model the single cell IC data as a hypergraph, where hyper edges in the graph can connect to multiple nodes. The input of the single cell IC data here is transformed into a hypergraph where each hyper edge connects one cell node is a blue node, and 2 genomic bin node is the red nodes here. So the model aims to predict the value of an entry. If your contact map initially is very sparse, we try to predict the values of these entries in the single cell high-C contact map using the rest of the contact map as input. So this setting shares some similarity with the self-supervised learning framework on graphs, where we mask portions of the graph, and then the neural net will be trained to recover the masked part with the rest of the graph as input. So the trained neural net is able to generate both the embeddings of the single cells according to the single cell high-C data based upon single cell 3D genomes, and also impute the sparse single cell high-C contact map. Just to show you one example very quickly, this is a data set from IPSL WST11. The first row is the initial single cell high-C contact map, the spatial organization of the genome. It's very sparse, as you can see, but through the imputations, we can fill in the blanks, and then these patterns starts to emerge. And you may wonder why we want to have these imputations, and the example on the right answers to the question, because the imputation will lead to much more reliable analysis of cell-to-cell variability of the 3D genome features. In this case, we're showing the so-called domain boundaries, where the score is the insulation score. If the score is low, it means the domain boundary is stronger. And at the bottom, you see that there is, these domain boundary you can observe from the pseudobulk level. You can see their cell-to-cell variability in individual cells. And if you don't do the imputation, then this analysis will be hindered, by the very noisy data. And what about complex tissues? And turns out that through this kind of ML methods, that we can use single cell high-C data to profile different cell types in complex tissues. And we apply Higashi method to the type of single cell high-C data called single-nucleus methyl 3C from Joe Acker's group. And note here that previously there was it was reported if you only use the single cell high-C part, you're not going to be able to separate some of the very fine scale cell types, like the subtypes in accessory neurons or inhibitor neurons. And we basically revisit this statement using a more advanced computational method. Turns out that we can indeed separate these fine scale neuronal subtypes. And then we look at also the connections between these fine scale subtypes between the 3D genome features and also the gene expressions from single cell RNA. And on these plots on the right hand side, I'm showing you two features. One is called single cell AB compartmentalization, and then the other is the single cell domain boundaries as reflected by insulation scores. And the scores, so each column is a single cell. You can see that these scores surrounding the marker genes of these particular cell types show distinct patterns as compared to the rest the other cell types. So this suggests that the connections between cell type specific spatial genome organization and also gene expression. Now, if we step back and to look at the cellular organization, spatial cellular organization in tissues, and we are interested in developing computational methods to model the cell identity based on both the intrinsic molecular features like gene expressions or these spatial organizations of the chromatins, and also the intercellular spatial organization in tissues. And this brings me to the main method I would like to introduce you today, what we call spice mix. So these are, as we all know, as we have heard from the previous two talks, there are emergent technologies to measure spatially resolved molecular features like gene expressions to study cellular diversity in situ in complex tissues. And our goal here is to have an integrated approach to consider both the gene expression that we are using the spatial transcriptome data and also the spatial organization of the cells. And this work was led by Ben Chidesta and Tianming Zhou, and then Xiaohu also contributed during the revision. And this paper was recently published as a cover on Nature Genetics. And even though on the cover it shows that it looks like the spice mix does look like some kind of spatial transcriptome, but our motivation of naming this tool is the following: if you put different spices with different proportions together, you get different flavors. And we think that cells may also work in a similar way, where you put different underlying factors with different weights and you get different cell types. And so this essentially brings to the explanation how the model works. And so the spice mix can operate on spatial transcriptome data set acquired by any method, and it can format the gene expression of the cell by gene matrix and model using the non-negative matrix factorization. In NMF, the expression profile of cell I, denoted by Y here, is assumed to be a weighted average of the metagenes, and the metagenes are shared by all cells and are collectively denoted by this capital M here. Each metagene is supposed to be capturing the expression profiles of one particular biological factors, such as cell types or differentiation trajectories, but it can also capture other factors like spatial, to reflect spatial organizations. The mixing weight of cells I here, the Xi, representing the contributions of different metagenes to cell I's observed expression value, Yi here. And then mixing weight here is very useful, and we consider them as the latent embeddings of each cell. Of course, one issue of NMF is that latent embeddings are typically assumed to be mutually independent. So the key method development here in SpiceMix is to relax this assumption, and then we allow the latent embeddings to be related to each other through a graphical model, the hidden Markov random field. And so I won't get into the details, but the outcome of the model, once we train the model on the data, the SpiceMix produces cell embeddings and capital S here, and then also the metagenes as well as their spatial affinities. You can see what are the metagenes that like to hang out together and what are the metagenes that don't like to hang out together. And so showing you some examples, we applied SpiceMix to various types of spatial transcriptome data, and here is the application to the mouse visual cortex from the star map data. And we first use SpiceMix. You can generate these from the latent embeddings from SpiceMix. You can do conventional clustering to identify cell types, but you can also look at their spatial patterns. And because of these embeddings are continuous, you can see the gradient changes along the tissue section. And on the right-hand side, on the top, I'm showing you the in situ cell type. These are the metagenes from the SpiceMix outcomes. You can see what are the metagenes, analysis of this in situ layer from the human various other methods as well as we described in the paper. But we did find that the layer 3 here, this layer is highlighted in this yellow arrow here. It looks like SpiceMix made a mistake initially. We thought that it may miss the mistake, but we took a closer look and found that this is actually something quite interesting. The axis here is perpendicular to the layer axis. Anatomically, the top layer here and then the right side, they're actually different. And this is the sulcus and this is the gyrus. This part is closer to the skull and to the skull. And then previous study shows that the gene expression in these two regions are distinct. And we found this is actually reflected in two metagenes from the outcome from SpiceMix. In particular, the expression of metagene A4 and metagene A5 supports the separations of these two regions. So basically here, what I'm showing you is that SpiceMix can identify these type of continuous gradients in spatial transcriptome data. And in particular here, this is the gyro-sulcal gradient. So to summarize, I mainly introduced the SpiceMix frameworks to model spatial transcriptome data, to infer complex cell identities, to review interpretable spatial metagenes, because it's a linear model, and so interpretability is natural, and the method can also uncover biological processes, which I didn't spend time to discuss. And we think that the model has a lot of potential to extend, and that's actually something what we are working on right now. First, we are really intrigued by the notion on how to model different spatial patterns in terms of their spatial and temporal changes. For example, during development, aging, or disease progression, if you collect samples in different time points, how to more integratively review for example, the metagenes, the spatial affinities, and then also the variations over time. And in addition, to connect to what I introduced to you at the very beginning, the spatial epigenome has different scales, and specifically how the epigenome is being shaped by cell microenvironments, and whether the epigenome may in turn modulate spatial organization of the cell. So we're actively thinking about these questions, including developing both experimental methods and computational approaches to advance our understanding of this multi-scale spatial epigenome architecture, which I think could be really useful for the human cell atlas community. So with that, I'd like to thank my group and also people who contribute to the work that I mentioned, Tianming, Ben, Shahu, Roch, and all others contributed, and also our funding sources. Thank you very much for your attention. I'm happy to take questions.
Thank you. Thank you. So thank you, Jian. That was very interesting. We have time for one quick question, I think, before we need to move on to the next session. Okay, can we have a question from Fabian coming.
Hi, we can do NMF also on the disassociated cells, or just take it from an atlas or something. Have you compared? So what would be the difference? Would you be able to sort of find then space-dependent component? How do you differentiate those two?
Oh, you mean comparing with conventional NMF?
Yeah.
Yes, so we actually compared evaluation both simulation, some real data, and certainly the differences varies, but we do see that the modeling of spatial factors explicitly does contribute to enhance the performance. And I think that was also reflecting some of the other recent works in literature. I know Barbara Engelhardt's group has also some recent work on that.
Makes sense. Thanks.
Okay, thank you. I'd like to thank all of our speakers from this session one more time. Thank you, everyone. It was interesting to see some themes emerging that maybe we can discuss more in the breakouts, like continuous modeling and relational modeling that doesn't bin the data such that it's kind of more flexible. So that'll be interesting to discuss. So I'd like to welcome Kerstin Meyer to host the next session.
Great. Thank you very much. I'm standing in for Ms. Hanifer this morning, and it's a great pleasure to introduce our next session, which is a new dimension to enable new questions. And it's a great pleasure to welcome Christina Leslie, who's from the Memorial Sloan Kettering Cancer Institute, and she's going to talk to us today about machine learning for regulatory genomics.
Great, thank you for the introduction. Thank you to the organizers for the invitation. Okay, so what do I mean by regulatory genomics? I mean assays that help us understand regulation of gene expression. So I want everyone to have a picture in their mind as we think about these data. There are distinguished elements in the genome, regulatory elements, enhancers and promoters, so both promoters near the transcription start site as well as enhancers that can be a megabase or more away. And there's 3D looping bringing enhancers close to promoters and enhancing expression of genes, right? So, and we have now 1D epigenomic assays as well as 3D epigenomic assays, as we just heard, for mapping these events. So especially chromatin accessibility that tells us where these distinguished accessible elements held together, held open by transcription factor binding, right? And we can now map the 3D organization, the contacts with chromosome conformation capture assays like Hi-C. So this is just some data from our 4D nucleome project with Danwei Huangfu, where we're seeing changes in accessibility in pancreatic differentiation and also the rewiring of 3D connectivity at the PDX one locus, which is an important diabetes gene. So for this meeting, what I want to talk about is that many of these assays are becoming available at single-cell level, especially single-cell ATAC-seq. And this introduces new computational and especially machine learning questions. Okay, so let's talk about single-cell ATAC. It's beautiful data. This is data from Rostro et al., bone marrow hematopoietic cell types. And you can see that at a pseudobulk level, it looks very much like bulk ATAC, but you can also see the dynamic range is 2 fragments, right? 0, 1, or 2. So this is very sparse data, and there are also very subtle batch effects. So if you do a standard single-cell RNA-like analysis where you create a matrix of cells by peaks and proceed with dimensionality reduction, graph construction, and manifold embedding, it looks like you're capturing beautiful structure, but in fact it's confounded by batch effects. Okay, so what we see here, the 2 green regions, they're both hematopoietic stem cells from different donors. So the idea of dealing with these challenging batch effects is that we have additional information in single-cell ATAC, which is the underlying genomic sequence. Okay, so the method we came up with for this is called cell space, and the idea is that we are going to learn a latent embedding of both DNA chimeras and cells into the same latent space. All right? So here's the idea that you can start with peaks, but it's even better to start with just genomic tiles. You sample a sequence from the genomic tile, and then you're going to use that sequence as a training example. It's represented implicitly by its chimeras, and you're going to learn embedding of chimeras in cells so that the implicit embedding of that sequence is pushed towards the cells in which it's open and away from cells in which it's closed. Okay? And at the end of training, you will have an embedding of all chimeras and all cells, and you can also embed additional information. So after the embedding, you can take a motif for a transcription factor binding motif, and you can embed that and then look at similarity between motifs and cells in the latent space. Okay, so when you do this, because you're using the sequence information, it has batch mitigating effects. So I'm taking the same data. We're not doing any explicit batch correction, but we're just embedding in the latent space and doing a UMAP, and I'm also embedding important hematopoietic transcription factor motifs. And this is just to show that the motifs for these transcription factors embed close to the cells in which they're active and open. And you can also see that the latent structure is recovering the hematopoietic hierarchy, so much so that you can do a Palantir analysis, start at the hematopoietic stem cell, and you can find all the most differentiated cell types as the endpoints. Okay? And again, this is the standard embedding, and you have this batch effect that's mitigated. Okay. We can also learn motif activities because we can embed motifs in the latent space. And in this case, this is a human cortex data set. It's actually a multiome data set, so we have RNA-seq, and we can see the expression patterns of the transcription factors. Okay, so we look at the cell space UMAP. There's also a nice deep learning approach called single-cell BASSET, which is also a sequence-informed embedding using a deep learning approach. So the embeddings are somewhat similar. Maybe this inhibitory neurons are not pulled away enough from glutamatergic neurons. But what I want you to see is that in cell space, the activity of the motifs correlates well with the cell types in which they are expressed. SUBAS, sometimes it's hit or miss, okay? So the deep learning approach may not always be capturing the sequence information. In particular, PAC6 is supposed to be expressed in only radial glia, and it's not active there. Okay, I should speed up. I want to just give one more example, since this is an atlas community. This is an atlas from Satpathy et al., looking at human samples, bone marrow, as well as PBMC. They did a bespoke batch correction. Here we're just embedding with cell space, no batch correction, and we're recovering a latent structure and the biological complexity properly. And this is the zoom in. If we re-embed the lymphocytes, we're seeing reasonable relationships between different cell types. Okay, so I wanted to tell you one more thing that you can do with single cell attack. So there is a notion that the co-accessibility of events can tell you about 3D connectivity, that you can infer regulatory interactions in 3D from co-accessibility. And there is a signal, but it's a faint signal, all right? So I think an unsupervised approach is going to be very difficult to really recover the contact map. But a supervised approach is more promising. So this is a method, Chromofold, we're about to submit. And the idea is that we use two kind of sources of information. 1D information, which is just the aggregated pseudobulk. We sample different numbers of cells to get different quality of pseudobulk as a data augmentation step. And we also use CTCF motif scores. And then we as a second input, we use co-accessibility over metacells, okay? And this is a convolutional neural net. We learn to predict these stripes, and we put the stripes together, okay? So a lightweight model only needs single cell attack, and it actually achieves state-of-the-art performance, even compared against methods that have a full sequence model, methods that use additional inputs like CTCF chip. So just to give an example of what you can do, you can learn this model in human data, training on matched single-cell attack and Hi-C, and you can generalize to a new genome, new cell types. So this is just showing the experimental Hi-C in germinal center B cells, as well as regulatory T cells at important loci, the BCL6 locus and Helios. And you can see that the structures that are predicted are pretty good, not perfect, but enough to give information about regulatory interactions that biologists might be interested in. Okay, final thing I want to tell you about. This is a model for multiome data, all right? So where we have single-cell attack and single-cell RNA in the same cells. And we and many other people are interested in gene regulatory models and mapping enhancers to genes and using that to interpret genetic variation. So the idea is very simple. You just have to do a number of things in the right way. So what we're doing is learning a regularized Poisson regression model in single cells. So we are at the gene level predicting gene expression from chromatin accessibility, okay? And we learn a regression model, and then we can interpret the regression model using Shapley values to identify cell type specific enhancers, all right? And here I'm just showing that there are other approaches for kind of converting single cell attack into imputed gene scores. And this approach called ScarLink outperforms them, outperforms Archer, and outperforms DORC scores for imputing these gene expression values. But what we're really interested in is interpreting the model and associating enhancers to genes in a cell type specific way. Okay, so what I'm showing you here is a model for CCR7. We're looking in PBMC data. I'm summarizing the pseudobulk accessibility and the distribution of RNA per cell type, and the regression model that we're learning is at the bottom. And then I can also do a Shapley score analysis. These are very efficient, approximate Shapley scores to get an importance per bin per cell type. And that's what the blue dots are. And this is a fine-mapped variant associated with asthma, and you can see that it is significant in specific cell types only for the prediction of the expression of CCR7 and its CD4 and CD8 T cells. Okay, so it can help interpret these values. One last point, when you have a gene-level regression model for many genes, you can use it to implement the idea of chromatin potential. So there's a very nice idea from Jason Buonrostro's lab that often in differentiation, chromatin accessibility at the locus precedes expression. And you can use this accessibility preceding expression as a directionality for trajectory analysis. So what we're doing is using predicted expression from our model versus actual expression to see that, that time arrow, okay? And doing maybe more robust implementation of chromatin potential. And this is some examples in the original papers, mouse skin share-seq data, and this is another example where we're finding trajectories. Okay, so I just want to summarize. Cell space is a Kamer-based embedding to learn latent structure in single-cell attack. It's engineered to scale very well. We can run it in 1 million cells. It mitigates batch effects without requiring an explicit batch correction. I also showed you 2 other methods quickly, chromofold, which predicts the high-C contact map using single-cell attack alone. I didn't show you all the things you can do with it. One thing you can do with it is use the model, fine-tune on high-C in a bulk tissue, and deconvolve chromatin interactions into cell types. And then ScarLink, I showed you a gene-level regression model for multiome data that allows, through Shapley analysis, enhancer to gene linking, and also provides a robust implementation of chromatin potential. Okay, I think I'm over time, so I just want to thank all the students who did the work, and many collaborators, and especially Kushal Day, who helped us with all the human genetics for the ScarLink project. And happy to take questions.
Great, thank you very much, Christina. Are there any questions? Avi.
Thanks, that was really nice. Actually, on the last part, I think that was ScarLink, right? And the application to our older share-seq data. At least in the way it looked on the slide, it seemed to be a lot more discontinuous than the original approach. Do you have a sense of where the discontinuity comes from? And do you think it's actually that's more reality, there's more of a jump, or it's more a limitation of how good the prediction is?
Okay, so you're talking about
when you had the two embeddings on the 2 sides. One was the old paper we had with Jason, and the other was the prediction from ScarLink.
Okay, so all we're doing is showing that you can get a vector field, and in chromatin potential, right, there is a lot of smoothing. It's not, I would say, yeah, as we all know. Oh, so it's not a full trajectory method. We actually have a different trajectory method that that is too new to show using neural ODEs, and I think that's a sort of more comprehensive approach. It's just that I think it gives some information, right? So I think it's just to give some arrows, right? But it's not giving full trajectories, right? And we tried, I think there's perhaps other things you could do with the predicted expression and observed expression. We tried a lot of approaches, but we thought, we're not developing a full trajectory method. We're just showing that a previous method can be done this way. Yeah.
Okay, thanks.
Yeah.
Great. I think we probably have time for just one more question. I think I'm going to go with online. Sam, if you have.
Hi, we've got a question from Dana Peer.
Okay.
Just to say, I'm a fan of cell space, but like every other method, sometimes it works better than other times. Is there a way to know when it's working well beyond our prior biological knowledge?
Yeah, it's a good question. I would say the failure mode, which means that you need some kind of tuning, is if everything is a blob, right? If it's not separating out. And there are different hyperparameters, like many methods. One that I didn't talk about is that the optimization is a margin-based loss. And sometimes if you have cells that are more similar to each other, like it's all T cells, right? Or an organoid model and it's all epithelial cells, sometimes you adjust that margin and then you start seeing structure. So I agree, it has a lot of nice properties that you can see latent structure without knowing the answer, but there is tuning and you can sort of pull things more away or have things more intermingled. It's true, yeah.
Great, fantastic. So if we could all thank Christina one more time. Great, and our next speaker is Paolo Cadinu from Boston's Children's Hospital and Harvard Medical School. Over to you.
Thank you for the introduction, and thank you for the organizer for the invitation. So today I'm going to present our effort towards understanding how the colonic tissue remodel in a very simple mouse model of inflammatory bowel disorder. And I'm going to do that by using MERFISH, an image-based technique for spatial transcriptomics. In particular, I'll guide you through how the very first MERFISH protocol that we developed in the gut enabled us to study the complex cellular and molecular organization of the healthy colon, as well as its remodeling in disease. And we quantified this remodeling, and what we found was that tissue niches and cell-cell interactions that were established in disease only. But what was really intriguing is that some of these transcriptomic signatures and cellular patterns that we see in all inflamed region of the mouse were also observing anti-TNF resistance ulcerative colitis patient, underscoring the fact that study this gut remodeling in mouse models is important for further our understanding in disease. And inflammatory bowel disorder, like ulcerative colitis and Crohn, are characterized by relapsing remitting inflammation of the GI tract. These are multifactorial disease that, as shown here in the cartoon, are characterized by defects in the mucus and the epithelial layer that favor the infiltration of bacteria and then in turn trigger a dysregulated immune response. But of course, there are a number of studies that shows that genetic background, environmental factors, and altered microbiome might contribute to increase the risk of developing IBD. What is clear is that this profound gut remodeling that involves host and microbiome in a non-uniform way is particularly hard to study with current technologies. In fact, a subset of these current technologies are incredibly good at providing large-scale tissue architecture picture, but they provide limited information about cellular and molecular details. This is the case of endoscopy and perhaps histology. On the other hand, you have genomic-based techniques like single-cell RNA-seq that provide in-depth cellular and molecular profile. But this comes at the expense of the spatial information. And what we wanted to do was to actually leverage new tech that can allow us to provide cellular and molecular details without losing the spatial information. Because this is important to answer specific biological questions. And we do so by using MERFISH, which stands for multiplex error-robust fluorescent in situ hybridization. The principle behind MERFISH are extremely simple. We assign optical barcodes to individual RNA, and then by performing sequential round of single molecule FISH, we're able to essentially read out these optical barcodes and then retrieve the RNA information. Because of the high throughput, high multiplexing, but most importantly the subcellular resolution, makes really MERFISH ideal for studying tissue remodeling. And we applied MERFISH to a very simple mouse model of inflammatory bowel disorder. In a beautiful collaboration with like Ronnie Novarsky's group, we leveraged dextrosodium sulfate, which is a detergent that is dissolved in the drinking water of the mice. And is used to essentially like induce colitis. We administer DSS for a certain number of days, and then we harvest the colonic tissue at different time points. So early stage in the disease, day 3, day 9, peak inflammation, and day 21, recovery. And we do so to then process this tissue according to our MERFISH protocol to profile about 1,000 genes. 100 to 200 of these, 1,000 genes are for cell type markers. The rest of the panel instead is like more of a discovery purpose, where we comprehensively target a number of like pathways and categories of genes that are fundamental either in homeostasis or in disease. And we do so to answer like very, very high level questions about like DSS-induced colitis. We wanted to understand like the cellular and molecular organization of the healthy colon, and of course like how does it change in response to like DSS. And most importantly, we wanted to understand like to what extent this like mouse model can inform us on like human IBD. At this point, you might wonder, like, okay, but how does like MERFISH actually look like in the colon? Well, we develop a protocol that is able to like retain the RNA integrity in a tissue that is like incredibly rich of RNases. And the result of this protocol is like shown in the images over here. You see on the left-hand side a slice of the distal part of the colon where each of the dots represented like one of the detected RNA and is colored by its identity. I'm showing like 8 of about thousands of RNA. And it was very clear from the beginning that the spatial localization of like the cell type markers shown on the zoom on the right-hand side has actually like the expected location. But MERFISH is a quantitative like technique. And therefore we like benchmark MERFISH against another technique, which is like bulk RNA-seq. And you see the results in this scatter plot over here, where each of the dots represent like one of the gene in our like 1,000 gene library. And then its abundance is measured either via bulk RNA-seq in the x-axis or via MERFISH in the y-axis. And you can see a beautiful linear correlation, suggesting that our protocol was able to actually like preserve the RNA integrity and reproduce the expected RNA abundance. At this point, by using like Baser, which is an algorithm that was like co-developed in collaboration with Peter Karchenko's lab, we were able to like assign these RNA to individual cells. And this allows us to profile about 1.4 million cells in 52 slices in 4 different time points of the disease. And you can see these like cells projected in either UMAP space on the left-hand side or in the 52 slices of this study. And they are colored by their major cell classes. And MERFISH was able to actually like recapitulate the expected diversity of these like cell classes. But of course we wanted to go deeper and we wanted to understand like this remodeling that is happening inside like each individual compartment. So on the left-hand side, you see the UMAP representation of all the epithelial cells. And then on the right-hand side, you see like 4 representative slices of the 4 different time points. Day 0 is like healthy, day 3 is like early stage inflammation, day 9 is peak inflammation, and day 21 is recovery. We were able to like reconstruct the full epithelial lineage going from like stem, goblet progenitors, goblet, mature colonocytes, enteroendocrine cells, M cells. But what I want you to notice is that the regular and symmetrical like epithelial layer that is present in the healthy colon leaves the space to a more distorted, non-uniform, and even absent epithelial layer in disease. And their composition is also changing. We see the emergence of a number of like inflammation-associated epithelia, as well as like repair-associated epithelia. And what was really fascinating is that pretty much all the fibroblasts in the mucosa and submucosal layer polarize in disease into like inflammation-associated fibroblasts, which are known to contribute to the disease. And of course, we look into like pretty much every other compartment. We look into the immune, we look into the endothelial compartment, enteric nervous system, and smooth muscle cells. And what we notice is that most, if not almost like all of these like compartments are actually responding to the disease. At this stage, we asked like a slightly different question. We wanted to understand and quantify this like spatial remodeling. In other words, are there tissue niches and cellular patterns that emerge in disease? To do that, we compute the cellular neighborhood. The idea behind cellular neighborhood is like exceedingly simple. So for each individual cell, we essentially look at the nearest neighbors. And we are able to then like generate a gigantic matrix of like cell-by-cell neighborhoods, which can be processed with like conventional single-cell pipeline. And you see the result in the UMAP down here, where each of the dots represents a cell, but now is colored by its neighborhood identity. And we can project back like the neighborhoods in space, and you see the emergence of actually like cellular patterns. But at this point, you might wonder, okay, but what these neighborhoods are actually capturing, what kind of features are they reporting on? Well, if we look at the cell composition of the neighborhoods that compose the healthy mucosa, these are actually reporting on very well-known cellular gradients and tissue architecture. In fact, if I look at the cell abundance as well as the cell composition shown in this like pie chart over here, we see essentially that like the epithelia, fibroblasts, immune, and smooth muscle cells are actually varying across the mucosal layer according to very well described like patterns. So for instance, mu1, which is like at the bottom of the crypt, is reporting on the stem cell niche. And you can see like, not just like because of the stem cells or goblet progenitors that are present in the epithelia, but also because like of the MRC1, CD163 macrophages, because of fibroblasts that are secreting Wnt. And vice versa, if I look at the top, now the epithelia is reporting on mature colonocytes, a different type of like goblet cells, fibroblasts that are secreting like BMP, and also like CD11C macrophages. Again, these neighborhoods are recapitulating like known cellular gradients. Now the question is, what kind of neighborhoods emerge in disease? Well, if I look at the slice here at the peak of inflammation, we clearly don't see any healthy neighborhood. What we instead see is disease-associated like neighborhoods that are characterized by the absence of epithelia and by, of course, like a significant presence of like immune cells, both of the adapted and then of like of both immune system. But what is also very, very interesting is that we don't see any healthy fibroblasts, but we see a number of inflammation-associated fibroblasts shown here as like IAFs. Some of them are just present in this like ulcerated tissue. And also what is pretty unique over here is that this luminal neighborhood. This is reporting on like excreted cells that are excreted into the lumen. And this is like a well-known hallmark of inflammation in colitis. And these cells are actually like, if you look at the composition, are mainly like different classes of neutrophils, as well as to a lesser extent like monocytes and macrophages. And just as a vignette, we wanted to understand like what was actually like driving these neutrophils into the lumen and other modes of communication that are established in disease. And when we look at the cell-cell communication, what we found was that not only neutrophils can recruit themselves like towards the lumen, but actually like inflammation-associated fibroblasts are secreting a number of chemokines that are actually driving these neutrophils towards the luminal boundary. But this cell-cell communication doesn't end here, and we see that inflammation-associated fibroblasts can actually respond to like IL-1 betas or IL-1 signaling like secreted by neutrophils, but also IAFs are able to secrete other growth factors that neutrophils can respond to. In general, what we see is that these like unique inflammation-associated fibroblasts to immune cell interaction may really shape actually the inflammation or the inflammatory like tissue in these like ulcerated like regions. And finally, what we wanted to understand a little bit was like how this mouse model can inform us on like human IBD. We look at two different aspects. The first one is that what we found was some of these IF states that we discover in mouse might be actually present in human UC. In fact, when we reanalyzed like single-cell RNA-seq data looking for the markers that actually like the IF were expressing in mouse, we see that these were expressed in like a specific like subset of cells in human UC. And the other aspect that we look into is about biomarker panels that have been actually like put forward in the last few years that they report on anti-TNF resistant ulcerative colitis in ulcerative colitis. Anti-TNF is like one of the major last line of treatment for UC. And what we found was that the mouse homolog of this like biomarker panel are actually reporting on ulcerated neighborhoods, suggesting that these biomarker panel are actually most likely reporting on ulcers. So to summarize, we essentially like define the biogeography of the healthy colon as well as like the inflamed colon. We define a number of like disease-associated neighborhoods, and most importantly, that we define their trajectory across the disease. And then some of these like disease-associated neighborhoods can inform us on like human IBD. I want to finish by thanking our lab member, in particular Jeff, and then of course like I should say a big thank to like Rosalind Shu, Evan Yang, Brianna Watson, and Christian Riardon Lockbaum for this current project, and there are other ongoing projects in the lab. And thank you for your attention.
Great. Thank you very much for that wonderful talk. Do we have any questions? Nadav. Also, could I ask the questioners to introduce themselves, please? We've got one question over here.
Hi, so I'm Nadav. I spoke before, but I wanted to ask about the kind of the spatial neighborhood analysis. Did you try to play with the different sizes of the environment and see how that affected the kind of like the gradients that you saw?
Yeah, this is a great question. Yes, we did. And I think the size of the cellular neighborhood is a parameter that you can tune depending on like what kind of feature you want to see, right? So you can go at one extreme where essentially your neighborhood is like one cell, and essentially what you're going to land onto is like your single cell RNA-seq data, or, and in that case, like, or let's say a neighborhood of like n equal 2, you want to see like the immediate like closest cells because you are interested in like, you know, proximity, like close communication. On the other end, you can look at like neighborhoods which are significantly larger because you're interested in like relatively large like architecture. So the answer is that yes, we look into it, and there is no specific parameter that is right or wrong. It depends on like what kind of features you're interested in.
Great. And then we had a question over there, Malt.
Thanks a lot for your interesting talk, Malte Lucan, Helmholtz Munich. I'm wondering, how did you select the 1,000 genes? Doing this, you mentioned kind of pathways and markers. Doing that by hand seems like a very time-taking process.
Well, the pathways and I would say like the discovery part, the beauty is that we weren't biased in the sense like we decided to go for like a comprehensive list of like cytokine and their receptors. We were like interested in like BMP and Wnt because these are known for, you know, to preserve like homeostasis in like epithelial and fibroblasts, so we include them all. And this is also because we were interested like downstream in understanding like cell-cell interaction. So you want to like make sure that when you include, for instance, like Wnt and BMP, then you're also including like frisols so that like you can understand a little bit more of like the interaction. For what concerned that like the actual like cell type markers, in that case we like leverage like literature to our, you know, to our best extent.
Great. I believe we have some questions online.
Thanks. Yes. So this is from Mark Elisua from CNAG. Thank you for an incredible talk. How did you define cell neighborhoods, and how would you adapt the definition to tissues that may be less dense?
The neighborhoods I showed very quickly were defined by simply like looking at the like, in our case, like 30 to 40 like nearest like cell. And how you define like in less dense tissue really depends. You can, you know, the easiest way is like just increasing like the size of the neighborhoods. So you might just like look into rather than the first like 40 nearest cell, you might say like, what's like what kind of cells do I see within a radius of like 20 microns or like 30 microns or 50 microns, and see like what kind of information you can retrieve from there.
Great. Okay. If we could thank Paolo one more time for a wonderful talk. And our last talk for this session comes from Caleb Lareau, and he joins us from Stanford University.
Great. Okay, well, thank you to Gary, Aviv, Donna, and the other organizers for the opportunity to share some of the work. I was really excited whenever I saw the session title, because this is a lot about how I think about my work in trying to define new dimensions from single-cell data to be able to answer new questions. And I've been predominantly doing this in the immune system, and I'll be sharing with you 2 vignettes of how we've been able to do exactly this. So just to audit the way that we typically do the first step of bioinformatics in single-cell data, recently we got a new single-cell experiment back. There was about 50 gigabytes of FASTQ data that came back, and the first thing that we did with it is we put it into Cell Ranger, and we got back our H5 counts matrix. And I think this is pretty typical for how we'll all typically analyze single-cell data. But the thing that I want to emphasize about this very initial transformation of the raw data that comes from these single-cell experiments is that you're actually compressing it by file size down to about 50 megabytes. This represents about 0.1% of the data by size from these really massively rich data sets. And the other 99.9% of our data is typically being discarded relatively quickly. And under this sort of auspices of where can we find new dimensions and where can we find new opportunities in the large data sets that we're generating, I want to posit that we actually have a lot of this already at our fingertips. And so what I'll be telling you about are two of our efforts that have spanned the last few years using somatic mitochondrial DNA mutations, as well as the presence of viral RNA molecules that were already sitting in the data and just required a little bit of extra juice to be able to then define new biological meaning from these. In other words, we're pulling out extra information from this other 99.9% of the data that come from our existing single-cell assays. So for the first part, again, I'll talk a little bit about mitochondrial DNA mutations. This has been work that's been going on for about the past 5 years. That was originally led by myself and Leif Ludwig when we were trainees with Vijay Sankaran and Aviv back in Boston. And so just to jump right into it, we were originally motivated by this goal of looking at mitochondrial DNA because of how the ATAC-seq chemistry, which I think needs a little introduction to this audience, really works, where you're using this hyperactive transposon to ultimately cut where accessible DNA is in the cell. And so the reason why most of us like ATAC-seq is because we can elucidate this really rich regulatory logic across different cell types, and we can see all the different regulatory events, as Christina nicely introduced a couple of talks ago. But the thing about the original ATAC-seq chemistry was about half of your data was restricted to mitochondrial DNA, being this really hyper-accessible bit of nucleic acid that's present in the cytosol. And so this was something that when I started my PhD, I was really motivated to try to figure out a way that we could get something out of this extra 50% of the ATAC-seq chemistry. And what we published in 2019 was this idea that mitochondrial DNA is actually somatically hypermutated. And so under this model, you can imagine that a cell will accumulate a somatic mutation in the mitochondrial genome. Typically, it's not functional, but it'll actually faithfully propagate that down to daughter cells. And so on the right here, I have a couple of fast facts where explicitly mitochondrial DNA is quite small, it's quite compact, but it's mutated at a much greater rate than the nucleus and ultimately is present at a higher copy number. And so you're really likely to detect it from a variety of single-cell assays. And so just to give a flavor for what this data looked like back in 2019, we isolated individual CD34 positive stem and progenitor cells, grew them up into colonies so we knew the true positive clonal annotations, and then profiled them with the chemistry at the time, SmartSeq2. And what we could find here is this really rich lexicon of somatic mitochondrial DNA mutations that faithfully reconstructed clonal processes that were occurring from a native human in vivo. So you can see the different clusters of columns indicated by the different colors are different colonies that were true positive events, and each individual column is a single cell. And so all of this blocky structure along the diagonal indicates different somatic mitochondrial DNA mutations that were marking these clonal events. And so since this original publication, we've scaled up both the technology, the bioinformatics, as well as some of the applications quite a bit. So nowadays, whenever one is interested in mitochondrial DNA lineage tracing, we recommend this assay called MT single-cell ATAC-seq. This is a modification of the commercial 10x Genomics ATAC-seq kit. And what this allows you to do is to faithfully profile both accessible chromatin as a measure of cell state, as well as the somatic mitochondrial DNA mutations. So in the hands of very capable and talented collaborators, Timo Ruckert and Chiara Romani in Berlin, they were able to apply MT single-cell ATAC-seq in the context of a question that they were quite interested in, which is how do natural killer cells respond to cytomegalovirus infection? And so explicitly, the experimental design is exceedingly simple. You derive natural killer cells using a combination of FACS and MAX-based enrichments from individuals that either have been infected or not infected with this virus. So no mouse models, no sort of genetic engineering to do this lineage tracing. But what we were able to find was really quite compelling, where if you then profile natural killer cells with this assay, you don't see just one blob of natural killer cells, you actually see a bunch of different pockets of these different natural killer cell populations. And in fact, whenever you layer on this extra layer of mitochondrial DNA mutations, what you could find is that these different subpopulations were actual clonal populations of adaptive natural killer cells. So this is super cool because natural killer cells are sort of thought to be this classic innate immune cell that doesn't have these adaptive properties. But what we're able to show with mitochondrial lineage tracing is in fact that some of these adaptive populations are clonally responding to this natural killer, to the cytomegalovirus. So ultimately, these were clonally heterogeneous populations in a very well-studied natural killer cell population that do not have distinct, say, markers by obvious flow cytometry panels, but ultimately discovered using single-cell sequencing. And the other thing that I'll mention about these populations now that we've been able to access these same donors is that they've been stable for 2 years in vivo. So again, these somatic mitochondrial DNA mutations were a feature of the data that until we took a look at our single cell with fresh eyes, something that really wasn't being appreciated in our analyses. And in the second part of this talk, I now want to share just briefly a vignette of how we've also been able to dig back into the data, this time in the context of therapeutically relevant T cells, where we've been able to identify evidence that there could be reactivation of a latent herpes virus. So this is work that I've been doing in Ansu Sapathi's lab at Stanford in collaboration with a very talented technician, Emma Yin, and we'll jump right into it. So as a second form of therapeutically relevant lymphocytes that we can now engineer in vitro, CAR T cells have been incredibly promising for their potential as anti-cancer killing cells. So explicitly, the way that you actually make CAR T cells is you'll derive T cells from a blood draw that you can imagine doing, and you'll actually engineer them in vitro by inserting a transgene that's going to hone the remarkable diversity of T cells down to where they'll recognize just a very specific antigen that will be present on the cancer cell surface. And in the process of engineering these cells, you're also going to grow them up into huge amounts and then reintroduce them back into patients to ultimately unleash this really profound anti-cancer killing response. And so the promise and excitement around these engineered cell therapies is that these have been incredibly efficacious in contexts where standard of care has been very limited. But some of the problems has actually been in the context of actually some of the toxicities associated with patients that have been receiving these therapies. And so explicitly, there's quite a bit of neurotoxicity that will actually incur from patients that are receiving CAR T cells. And so one of the major goals of my work at Stanford has really been to try to understand, are there potential mechanisms of neurotoxicity that we can ultimately attribute using genomic sequencing data, and then ultimately potentially make these therapies safer for future patients. And so one of the things that we were able to find just by reviewing some of the press releases and some of the footnotes in clinical trials is that you would have these major releases from FDA-approved products where they would recurrently note the possibility of what was termed HHV6 encephalitis. So explicitly, this is a swelling of the brain that can be lethal that's attributed to the human herpes virus 6 pathogen. And ultimately, it was something that you would see in a major press release from someone like Gilead that has an FDA-approved product. You would also see it from startups like CRISPR Therapeutics that would note the occurrence of HHV6 encephalitis from patients receiving their newer therapies. And perhaps most soberly, there was a press release from Novartis that has Kymriah, one of the major FDA-approved products, where they actually noted the instance of a 4-year-old girl with refractory ALL. The CAR T cells were working incredibly effectively. Her cancer was in full remission, but she ultimately tested positive for HHV6B and died due to encephalitis. Again, her death attributed to this reactivation and this neurotoxicity associated with the virus. And so overall, about 1 in 40 patients that are receiving CAR T cells will actually go on to develop this form of neurotoxicity that can be attributed to this viral event. And so we've wanted to ask the question, could we actually pinpoint where this virus was coming from in the context of individuals receiving this cell therapy? And so this is a particularly interesting pathophysiology because a human herpes virus is actually endemic. So if you didn't know this, I have some bad news. You likely have been infected with HHV6 at some point in your life. Here I'm showing a cumulative incidence plot where in the first 2 years of life, anywhere from 90 to 95% of individuals will be infected with this virus. So you've been infected, but typically your immune system is ultimately going to control the primary infection. So there's the initial lytic phase, but ultimately a subset of your cells will actually harbor this virus in what's termed long-term latency. And due to a variety of factors that we don't fully understand, there can be a reactivation of this viral entity that ultimately then causes this encephalitis phenotype in a subset of patients. And so the way that we're able to actually pinpoint what we think is a potential source of this virus is through a reanalysis of atlas-level sequencing data. So explicitly, you can imagine that whenever you profile your cells using single-cell RNA-seq as an example, you would have both host RNA expression that you're typically carrying around in your counts matrix, but you'll also have viral RNA that you're probably not considering anything with right now. And so what we were able to do is actually take 3 different cohorts spanning different studies that explicitly profiled both CAR T infusion products as well as peripheral blood mononuclear cells from patients receiving these. And what we're able to find is actually the incidence of CAR-positive, HHV6-positive cells in vivo. So explicitly on the right here, I'm showing a heat map of different cells along the rows and different viral genes along the columns. And you can see that whenever there are cells that are HHV6 positive, they're actually across a diversity of transcripts, and they're actually quite high. And so explicitly, whenever we found this in the data, we actually were able to infer and ultimately show using additional in vitro data that there is, at least in some patients, the possibility of HHV6 reactivation coming from the process of manufacturing these therapeutically relevant CAR T cells. And so explicitly for one of these patients where we had single cell data, we're able to actually map a instance of the time course of the single cell profiling onto their clinical course. And this individual did in fact actually test positive for HHV6 via sort of a clinical, standard clinical PCR. And ultimately this problem was mitigated with the treatment of foscarnet. And so explicitly what I've told you about in the second vignette is that we're able to reanalyze other people's single-cell RNA-seq data to ultimately find something that we think is potentially clinically actionable to ultimately make these CAR T-cell therapies safer. So just to take home what I try to share with you over the course of these two vignettes, again, this very, very initial transformation that we make to the data is really vastly compressing the amount of information that we're gleaning from these single-cell experiments. And what I told you in these two vignettes are how we've been able to infer different populations of lymphocytes that would typically not be considered if you didn't have these additional information. Explicitly, the fact that you can have somatic events in the mitochondrial DNA, as well as you can have reactivation of latent human herpes virus 6. And so my suggestion for the consortium is that certainly if I were to take only 0.1% of the data, I would certainly want the counts matrix. It's absolutely the most information rich. But especially as we start to mass data at scale, I think we should periodically reconsider what else may be in this other 99.9% of the data and how we could potentially actionize this to make new biological discoveries. So I try to give a shout out to the individuals that I did the work with as I go, because I wanted to save the last slide for some exciting news. So as of 2 months from now, I will be moving to Memorial Sloan Kettering, where I'll be starting my own group in the Computational and Systems Biology program, the same program that houses Christina and Donna. And so I will be trying to recruit my lab from 0 people. So if you would like to join, please reach out. Thank you very much for your time.
Thank you very much for a fascinating talk. We have one question over here.
That's awesome. Bruce Arno, Cincinnati. So you've been following EBV reactivation and the use of ART to
absolutely.
suppress it, and so you think HHV6 is also integrated into a repetitive DNA element and getting endogenous retroviral transcribed that you could suppress it?
Yeah, I think there are effective antiretrovirals if you can catch it. I think genomics is a perfect place because if it's happening, you can just detect it in the nucleic acids. In that specific context, EBV has a different latency than HHV6. So HHV6 does typically reside in T cells, EBV in typically B cells. So depending on sort of the treatment regimen, you could have different stressors to different cells that lead to different viruses reactivating, I think.
Not the reactivation, but treatment of a reactivation using antiretroviral therapy to try and suppress it. Is that?
I would probably have to follow up with you on the discussion.
It's working in EBV reactivation, and for reasons that are unclear. And so it could apply here.
Yeah, I think the thing that we were able to show from this is that we pinpointed the source, and so now we're hoping that HHV6 was typically in the CAR T setting, like a super tertiary differential diagnosis. Now we've been able to molecularly more pinpoint it at what it could be causing, and so I think the fact that there are therapeutically relevant options, we just want to now bring that to physicians' mind that this could be treated if caught carefully. So thank you for your question.
I was also wondering, in your NK experiment, you have those clonal populations that are clearly different somatically. But do they, you have ataxic data, so do they show different activation of different genes, or are they mostly homogeneous otherwise?
Yeah, no, so just to clarify that, UMAP is actually derived exclusively from the ataxic data. So the fact that they're differentially clustering does indeed point to them having distinct epigenomes that also share these clonal somatic mutations. So they definitely have completely different epigenomes. There's definitely some functional relevance to that, but that's still something that we're trying to fully appreciate now.
I think we're at time, so I'm now going to hand back over to John for closing it out, but let's just thank Caleb one more time.
Thanks, everyone. We'll have about a 35-minute break before the breakout session, so please enjoy refreshments and so forth. We'll have staff circulating about 5 minutes before the breakout session begins, and we'll ring a bell and so forth, so you will know that they are about to begin. Then we'll have lunch, as usual, about 12:30 for about an hour and a half, and then please be back in this room at 2 o'clock for our keynote address. And then later in the afternoon, we'll have another round of lightning sessions, another plenary session, and then close the day. So thank you very much. Enjoy the break and enjoy the breakouts.